Hey everybody. This short lecture deals with the introduction to the cells. In here we'll be talking about the different kinds of cells and how cells were discovered. And that's where we're going to start. Alright, the cell theory is actually composed of three parts, and it was formed primarily by these first two individuals, Robert Hooke and Anton van Leeuwenhoek. And it's consisted of three parts that were all contributed by different scientists throughout the years. Robert Hooke is credited with being the first person to describe cells. As he made his first microscope, he looked at little pieces of cork and described the rooms as cells because they looked like the rooms that monks lived in monasteries. Later, Anton van Leeuwenhoek was the first one to actually describe living cells. And he started seeing these cells, he called them in pond water, called little animals because they were moving around. Now, as scientists progressed throughout the years, they picked up on three things about all cells. Okay, first part is whether you're a plant or an animal, all living things are composed of cells. The botanists who study plants and the zoologists who study animals made those discoveries independently, and then they combined them together into one comprehensive part of the cell theory. The second part is that all cells, the cell is the basic structure, basic units of structure and function. In other words, you can't have anything smaller than a cell and still be alive. A cell is the smallest part of anything, or smallest component of a living thing that can be considered alive. And the third part, came from the idea that, cells can only come from other cells, which leads to a whole new argument: where did the first cells come from? But we'll discuss that much later in this course. Variety of sizes. Okay, some can be seen without a microscope, but most are going to need magnification. If you look at my chart over here that I borrowed from one of the materials we use for this class, some cells, like a chicken egg, which is a single cell, can be seen without a microscope. And even some of these others that we look at, like frog eggs, and some, one-celled organisms you might see as little dots, can be seen without a microscope or the unaided eye. Now, what limits the cell size is a combination of several things. The biggest thing is the ratio of the surface area to volume. So long as that ratio, if there's a whole lot of surface area to the volume, then the cell can actually exchange nutrients and materials fast enough here to support the entire inside of the cell. If the cell gets too big, like in that old movie called The Blob, where they basically took a picture of an amoeba having it swallow up people in cars, a cell couldn't exist that large because the amount of surface area available can't support that huge volume. And that's because if you look at a cube, probably the easiest thing to measure the surface area and volume of, when you increase the volume of a cube, that increases with the cube. So remember, to calculate the volume of a box, you would take the length times the width times the height. That gives you your volume. So if the box is a square on each side, you would take length times width times height. So if you had a box that was 3 millimeters on each side, it would be 3 times 3 times 3, or 27 millimeters cubed. But the surface area would be only 3 times 3, which would be 9 times 6. So that would be 54. In this case, the surface area to volume ratio would have been 54 to 27, or 2 to 1. If that surface area to volume ratio increases more, it's going to exchange nutrients much better, much more efficiently. And materials can be, like food and waste materials, can be exchanged more rapidly. A variety of shapes and sizes. The shape of the internal structure here determines what it's going to do. For example, if a cell had a large number of organelles designed for the breakdown of alcohol, a biologist might assume that function of the cell, was to rid the body of alcohol, and that's pretty much the truth. Because we always know that the structure relates to the function. Okay, and besides certain organelles that would detoxify the blood, there's lots of other functions here. The cell membrane, remember, is selectively permeable, so that means it allows some materials to pass and keep others out. Well, there's certain features that all cells are going to have. For example, all cells will have a cell membrane. also called a plasma membrane, which separates the interior of the cell from the outside. And this membrane is selectively permeable, which means that it's going to allow some materials to pass through and yet keep others out. All cells will also have cytosol. Now, a little hint to this: the prefix cyto means cell, sol referring to a solution. So this is the liquid portion of the inside of the cell, where you're going to contain all the dissolved solutes that are present. Whereas cytoplasm still refers to the cell. This refers to the liquid portion, the dissolved materials, as well as all the organelles that are present. And all cells are going to have ribosomes. Ribosomes are the protein factories of the cell. And it doesn't matter what kind of cell you've got, they will all have a ribosome. And all cells will have a genetic material, in this case the DNA. It will be contained within the nucleus of the cell. Now, not all cells will have a nucleus, as we'll see later on. Now, there are two types of cells you need to be aware of: prokaryotes and eukaryotes. Prokaryotes show up earlier in the fossil history, so they are technically considered the older of the two kinds of cells. They do not have a nucleus or membrane-bound organelles, like in this picture right here. While the prokaryotic cell does have DNA right in here, it is not confined within a covering. This is an area called the nucleoid, just basically where the single strand of circular DNA is looped around. And this picture right here shows a bacterial cell, which would be an example of a prokaryote. And here's the actual electron micrograph from a transmission electron microscope. Okay, they both have, the nucleoid material just in a central region. There are no membrane-bound organelles. Some will have a capsule, which covers the outside of the cell. Okay, and some will have a flagella right here. Okay, all prokaryotes are extremely small. Compared to a eukaryotic cell, these are almost even microscopic to their standards. Eukaryotic cells, like this very colorful diagram of a cell here, eukaryotic cell, basically are differentiated because they have a true nucleus. They actually have the DNA enclosed in an area right in the center. And the other cell organelles